In this chapter, we plunge into the heart of what drives living systems, and that is gene expression. So we've already talked about genes and what they are. They're regions of DNA on a chromosome that code for something, but the thing that they actually code for is the production of protein. And protein is a very essential thing to make in the cell because protein's roles are so diverse. Protein can be used for structural substance. It can be used as a enzyme. It can be used as a channel or a receptor. So oftentimes things that are built in the cell are built because there are different proteins around that can build them. So if the cell is able to make protein, then it can use those proteins to build any of the other types of things that are required, whether it be DNA, or any kind of nucleic acid, or a lipid, or any of the other types of macromolecules. So genes ultimately code for the production of protein. Genes don't generally build anything else. They don't build nucleic acids. They don't build lipids. Genes code the information to build proteins, and then proteins are the thing that's used to build all the other stuff that's required. So how is it that we get from a series of bases on a chromosome to an actual fully functional protein that can be used by the cell? Well, that's a little bit about the subject of this chapter, and it really is what we call the central dogma of biology, because it's basically the main idea, the main core thing that's happening that all life is centered around is the ability to express genes and use them to make protein. The second half of this lecture then will turn our attention to some of the applications of biotechnology that are around today. Okay, so how it works, essentially, here we have a picture of a cell. The genome is housed in the nucleus. This little dark structure here, this would be the nucleolus. We never really talked about this, but this is where RNA, a certain type of nucleic acid known as rRNA is made. In either case, in that genome, there's DNA, and the DNA will get copied to form RNA. We said that DNA, as housed in the genome, again, is like a library with many, many books of instruction manuals that the cell can reference should it need to build something. RNA is like a photocopy from that library, and then RNA is going to go out into the cell and actually be doing stuff. Now, there's three main types of RNA. There's several other different types, but the big ones are mRNA, rRNA, and tRNA. mRNA stands for messenger RNA, and messenger RNA is the photocopy of DNA, which provides the instructions for how to build a protein. Okay, it has on it the instructions of how amino acids need to be assembled, what order they need to be assembled to make any particular protein. rRNA is a special type of RNA that forms ribosomes. So ribosomes, we mentioned earlier, are an organelle in the cell, and they are the site of protein synthesis, and they are actually made in part from these strands of rRNA, and rRNA is also made from DNA. Okay, tRNA then, this is sort of I like to think about it as the bridge between the genotype and the phenotype, because these special molecules, they have a portion of them that can bind to nucleic acid. They also have a portion of them that can bind to amino acids. So it's kind of the link between nucleic acids and then the subsequent protein that is built. And we'll see a little bit more of this in action here a little bit later. Regardless of which type of RNA is made, the process by which DNA is converted into a smaller strand of RNA is known as transcription. So these are all called RNA transcripts because basically they're just little small copies from the DNA genome. Now, rRNA is, just for reference sake, it's usually single-stranded, not double-stranded. Remember, DNA is shaped as a double helix, two strands wrapping around one another, think about it as like a twisted ladder. Okay, that's the double helix. RNA is more single-stranded. It can fold into more complex shapes. Generally has a more diverse array of roles. It doesn't just store information. It actually can be used to make ribosomes, for example. And also rRNA. rRNA has not the base thymine in it, but instead the base uracil. So if you're ever looking at a sequence of bases and you see U's and not T's, we know we're talking about RNA. Okay, a picture of what this looks like is shown here. So essentially here we have a chromosome. And if we were to look at some segment of DNA along that chromosome, in order for transcription to occur, the DNA has to separate. The two strands have to separate to allow some space. Generally what happens is that a protein known as the RNA polymerase, so it's actually specifically an enzyme, because it's something that actually puts things together or takes things apart. That's usually what enzymes do. Well, this is something that puts things together. The name of the enzyme is RNA polymerase, meaning it makes polymers of RNA. Okay, so what this protein does is it binds to the template DNA, and it just goes along and reads the template strand, and it will add complementary bases as it goes along. So, for example, here we see a T. So it's going to add a complementary base, which would be A. Remember, A is always bind with T's, and vice versa, and C's always bind with G's, and vice versa. Okay, in this scenario, though, we see there's an A on the template strand, so it normally would add T if it was DNA, but because it's RNA, there is no T, it's U instead, so that's why the U is added. And here's another T, so an A is added. Here's another A, so a U is added. Here's a C, so it adds the complementary G. Here's a G, so it adds the complementary C. And using that complementary base pairing rule, it can create a transcript which is complementary to the DNA template. Okay, that's generally speaking the process of transcription. Now, it gets very complex because where this polymerase binds and when it binds and what recruits it to come and then bind so that it then makes the gene, that's all very highly regulated, much beyond the scope of this class. Cells are not always expressing their genes, meaning cells are not always copying their genes and sending the transcripts out to build protein. It might be that one particular scenario requires a certain protein being built, so on some place some gene will be transcribed. Another time later, a different environmental condition will require that a different gene is made, and so not all genes are expressed at the same time. Okay, then what needs to happen is the protein needs to be made, and that process is called translation. In translation, because it's translating the code that's held in RNA, specifically messenger RNA, and it's translating that code into a different type of thing, which is a sequence of amino acids. And a sequence of amino acids is a protein. Okay, the sequence of bases on mRNA corresponds to the addition of various amino acids, and the genetic code is the code that is the link between the two. Okay, so the genetic code looks something like this, where it's arranged in 3-letter sequences. Those 3-letter sequences are called codons. So let's say that this was a messenger RNA transcript that was made. Every 3 letters would be a codon. And each one of those 3 letters, if we looked up the code, we could see what amino acid it would code for. So for example, this codon here is UAA. So the first letter is U. So I would come over here to the first letter and find U. So that would mean I'm going to be somewhere in this column. The next letter is A. So that would be second letter is A. So that means I'm somewhere in this box. And then the third letter is also an A. So that would mean that codon is actually going to be what's known as a stop codon. It would actually signal the completion of making the protein. So I guess this isn't really a great example. Another example would be like UGG. That would be UGG. So that would be right here. That would code for the amino acid, the addition of the amino acid tryptophan. Okay. Now the reason why these codes exist is going to be related to the nature of tRNA molecules. But before I talk about those, I just want to make mention of the fact that all of these letters here are basically abbreviations for the different amino acids that we had already talked about. We don't have to memorize all of the different amino acids, but just want you to know when you're seeing these, this means that there's some codon that's coding for an amino acid, and there's some times where multiple codons, like for example proline, which is this one right here. Proline has 4 different codons that would code for the addition of proline. Okay. Okay, so it's the sequence of amino acids that determine how a protein will eventually behave. Remember, proteins are just long chains of amino acids, and based upon the various chemistry of those amino acids, it will cause a change in the structure, and it will be the structure that actually determines its function. So some amino acids are positively charged, some are negatively charged, some are polar, some are non-polar, and depending on the sequence, it will lead to the formation of a long chain of amino acids that behaves in some way. Maybe it might lead to the production of a long chain of amino acids that acts like insulin and recruits or stimulates the uptake of glucose out of the blood. Okay, the process of translation of actually building the protein, this happens at the ribosome. In this illustration, the ribosome is this green thing, and how it works is that mRNA is going to be fed through the ribosome. The ribosome has different sites on it that allow, one of the sites allows a molecule of tRNA to enter, and the tRNA is what we're seeing here as these little molecules. Now, tRNA is the reason why the genetic code exists. tRNA is the reason why each codon on an mRNA leads to the addition of a particular amino acid, because on a tRNA molecule, there's this thing that's called an anticodon. And an anticodon will be attracted to and bind to its complementary codon. So for example, if the codon on this particular part of mRNA right here was like CGU, the anticodon would be GCA. It would be whatever is complementary to the codon. Okay, so all that to say, when the mRNA reaches to the point of the ribosome where the codon is now exposed, there will be a whole bunch of tRNA molecules in the cytosol surrounding this ribosome, and they will very quickly be whichever codon, when this codon has been made accessible, whichever tRNA bears the complementary anticodon will then be attracted and be pulled in and attach to the codon. So there's a lot of different tRNA molecules that all have different anticodons, and depending on the anticodon that a tRNA molecule has, it also has a specific amino acid that it attaches to. So you can see that this really is the link here between the nucleic acid part and then the amino acid part. So as this ribbon of messenger RNA is fed through, it leads to a sequence of amino acids being linked together, and that's what we're seeing here. There's literally a long chain of amino acids being formed as this messenger RNA is read through the ribosome, because as each one of these codons becomes accessible, the complementary tRNA will bind to it. It will bring with it its amino acids, and the amino acids, once they're close together in proximity, there's a bond that happens where they actually will fuse together. And then as they fuse together, it forms this long chain which emerges. Okay, there's a really great video that shows this. I'll just play it here quick.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
In this next part, we're going to look at ways the various processes and biological systems are utilized as tools in society. One such application of this is in what's known as molecular cloning. This is a way that we can actually make a lot of a particular molecule, and it oftentimes involves using bacteria. Bacteria are unique because they have their genome, but in addition to that, they have these little accessory rings of DNA called plasmids. I like to think of these plasmids as conferring some sort of like special trait or quality to the bacteria. For example, oftentimes if a bacterium is going to develop antibiotic resistance, the gene that codes for whatever it is that makes them resistant is occasionally found in a plasmid. But in either case, what we can do is we can actually insert a plasmid into a bacterial cell, and let's say there's a particular gene of interest that we want to express. We can put that gene into a plasmid, put the plasmid into the bacterial cell, and then the bacteria will grow and reproduce, and the bacteria should make whatever product is coded for by that gene. So an example of how this is done is basically what can happen is we can take a plasmid and we can cut it. When we cut it, depending on which enzyme we use to cut the DNA, it can sometimes cut the double-stranded DNA like this so that there are exposed bases. These are known as sticky ends because they allow one to insert another segment of DNA that will then attach to those ends. If we just cut it straight across, it would be difficult to add insert a new strand of DNA in there because there wouldn't really be anything for it to bind to. But when it does cut in this fashion where it's allowing a couple of exposed bases, then what we can do is we can cut the plasmid, figure out our gene of interest, and then put it into the ring to close the whole thing up again, and then put that into a bacterial cell. Now, a common example of how this is done is in the production of insulin. So insulin is a human gene that is put into a plasmid and then put into a bacterial cell. And the bacteria are grown and multiply and multiply and multiply, and as long as they're fed and they have the proper nutrients, they'll grow and they should produce insulin, human insulin, not any kind of bacterial protein, but a human insulin, because the human gene is now inside of the cell. So this is showing a facility here where there's lots of these vats of bacteria growing, and they're producing insulin. The insulin can then be extracted and purified and bottled up and sold to people who need insulin. It's a very efficient way of making a lot of a product. Another example would be reproductive cloning. So this isn't just to mass produce any particular molecule. This would be to actually clone an entire organism. How this has been done in the past is that an egg cell is taken from a donor, the nucleus is sucked out, and then diploid cells from another organism are cultured, and the nucleus of those diploid cells is then put into the egg cell, like this. So this is unique because an egg cell is haploid. The nucleus that is being removed would be haploid, and then instead a diploid nucleus is being inserted, and that nucleus will be a clone of the genome of this donor organism. So then now we actually have an egg cell with a diploid nucleus that is an exact copy of any of the cells, the diploid cells in this animal. This almost is then like a fertilized egg, except it didn't get fertilized, it just had a diploid nucleus put into it. That cell can then be put into a surrogate mother, and then it basically just develops like any other fertilized egg would develop. Then it would give birth to the offspring, and the offspring are an identical clone to the original nuclear donor, the one that donated the nucleus. Now, what is the significance or the relevance of this? I'm not exactly sure. I know that dogs can be cloned, and sometimes dogs are cloned if a particular owner really, really loves their dog and they never want it to perish. They can just clone a new one. This is something that's being done in various countries of the world. Apart from that, I'm not exactly sure what the importance of this technique is, other than to say that we're able to clone an animal. Okay, genetic engineering refers to putting genes into an organism's genome, or taking genes out of an organism's genome, or just changing genes in an organism's genome. One example of this is, or a lot of examples of this revolve around this particular organism. This is a type of jellyfish that produces a protein known as GFP, which stands for green fluorescent protein, and it's basically a protein that glows. This is a special protein that's produced in a variety of organisms. It originally was discovered in the jellyfish, but it's also found in sea anemones and other types of organisms. This is used a lot in research because the fact that it glows, it makes it really easy to determine if some organism has received the gene or not. So oftentimes if people are doing genetic research like this, they'll put some sort of gene of interest and attach it to the gene that has GFP, and then as they're going about and trying to put genes into and out of organisms, they'll know that they have a successful, they've been successful if it glows. So it's like a nice label or a nice marker to help one figure out what's happening with the genes that they're introducing or taking out. An example of this, though, would be glow-in-the-dark mice. You can actually get glow-in-the-dark mice, or even fish that glow. Glowfish, basically all that's done is, at one point in time, the embryo of one of these organisms was treated with this GFP gene, and then all of the cells that developed from that original cell would then be copies of it, so they would be proliferating. And if you successfully create one mouse and you breed it with other mice, you can actually then pull out, through the way that you're breeding the mice, the gene in the population, so you can actually come up with a whole colony of mice that have that gene, or a whole colony of glow-in-the-dark mice that will continue to pass the gene on to their offspring, because they're true breeding for the trait. It's the only allele possible in the population. Another application of this would be in crops. So genetically modified, like GMO crops would be crops that have a genetic resistance to certain sprays, and so the spray can be applied, and all of the weeds are not genetically modified, so they are killed by the spray. But then the plants that have the gene introduced that makes them resistant to the chemical, they can actually survive. So it kills all the plants and except for the crops. Another interesting way that this is done is BT crops. Maybe you've heard of BT before. There's like BT cotton and I think BT wheat and some other things. But what BT crops are is they have a gene introduced into the genome originally taken from an organism called Bacillus thuringiensis. And this organism here makes a crystalline molecule that is actually an insecticide. It's like a natural insecticide. And so if we take that gene that codes for the natural insecticide and we insert it into the genome of a particular crop, as that crop grows and reproduces, it passes the gene on to its offspring. So now we have a whole colony of crops that naturally produce this insecticide. And the insecticide, in theory, is a natural molecule because it's made naturally by bacteria. A recent tool that's been developed, which has made genetic engineering much easier, I guess, is this system called the CRISPR-Cas9 system. And this is unique because the CRISPR-Cas9 system is basically a way to pinpoint specific spots on the genome, but not only pinpoint specific spots on the genome, but also then pinpoint specific actions to be done. So this is a powerful tool because on this particular complex here, the Cas9 complex, there's what's known as a guide RNA. And the guide RNA can be modified quite easily. And basically, when it's introduced into a cell, it will bind to its complementary sequence on the genome. And so if we can easily change the guide RNA, you can easily change where it is that this Cas9 complex is going to attach. So if you know whatever particular gene of interest you're hoping to modify, just modify the guide RNA so that it finds it, and then you can change the Cas9 complex to either, when it does bind, you can have it cut the genome there, you can have it introduce a mutation there, you could have it introduced a small fragment of DNA there, or what have you. So it's become quite complex, but at the end of the day, it's made it a lot easier to knock out genes or introduce new genes or turn genes off, delete genes out of a genome, which has a lot of varying applications, right? On the one hand, there are a lot of great things that could become of this, because we could start to, for example, knock out genes in pathogenic organisms that cause disease. So we could knock out genes in mosquitoes that make them malaria resistant, so they don't carry the parasite that causes malaria. We could make it such that Asian carp only produce male offspring, and then after several generations, then there will be no females for them to mate with, and then the invasive species will die off. But on the flip side, this has also been raised some concern, because a tool such as this could be used, for example, in bioterrorism. It could be used to engineer a virus. Some people think that, are worried about the smallpox virus coming back into the population, because it's, it was been eradicated, but it could be genetically engineered and re-released into the population, and then it would spread. So, as with all technology, with great power comes great responsibility, and all powerful technology out there has the possibility to make life better, and also the possibility to make life worse, depending on the intentionality of its wielders. So what we can do is we can hope and pray for the best. Okay, hope and pray for the best, and trust in God as our provider and our Savior, and for our own sakes seek to be responsible in the arenas that He has put us, and seek to be responsible with the tools that He has given us, and then trust Him. Okay, another example of these types of applications for biotechnology would be genetic therapies. So this is something that I actually did a little bit of work on here in the lab that I worked at the U of M. Specifically, we were studying muscular dystrophy, and muscular dystrophy results, it's a genetic disorder where there's a protein, a mutation in the gene that codes for the protein dystrophin. And dystrophin is considered to be somewhat of like a molecular shock absorber for your muscles, dampening the force of all of that muscular contraction so that it doesn't cause damage to cells. And so people that don't have dystrophin, they don't have that shock absorber, and so they are more subject to incurring damage in their muscles. So the thought here then was, well, if it's all that they're missing is the gene that codes for dystrophin, why can't we just package the gene that causes dystrophin into a virus, infect the host with that virus, and that virus will spread throughout the organism, and it will deliver into all of its cells the nucleic acid sequence required to make the dystrophin protein. Now, it turns out this is quite a bit harder than it sounds for a variety of reasons. One is that you can only load, viruses are generally pretty small, so you can only load, you're limited on how big the gene can be. So in our lab, we were actually using a micro dystrophin gene, a gene that coded something that was kind of like the dystrophin protein, but it was a smaller version of it, just because we were limited on size. Another limitation is that you have to use a virus that isn't going to be pathogenic, and it's not going to cause human disease. So that's tricky. The other part of it is that there are a lot of complex mechanisms at play that determine how the gene is turned on and how the gene is turned off. And so some of those other complex mechanisms, generally there might not be enough room to include those into this gene that's added. So it becomes a little finicky. But that is the thought, that for genetic disorders, it might be the case that one day we're able to better cure those disorders by introducing viruses which add healthy versions of the DNA into host cells. That concludes our lecture here on gene expression and biotechnology.